Welcome everyone. Today I'm gonna discuss about something about biotechnology. Today I'm gonna discuss about define the biotechnology, describe applications of the biotechnology, describe the uses of biotechnology in daily life, define bacterium, explain the process of DNA replication, describe how DNA is copied. Biotechnologies. Literally, the study of the tools from living the things. What is the biotechnology? Biotechnology is branches of science in which living things are used to make new product. It is the use of living organization or process to make product useful for mankind. History has been existing since centuries, began with the first actions of human on life for his welfare. It started when people modified microorganisms into the needs of humanity. It passed biotechnology used yeast cells to the farm alcoholic drinks and the bacteria to make cheese and yogurt. Term coined by a Hungarian engineer Karl Archi. Modern biotechnology started in California in 1970. Before 1971, the term biotechnology was used primarily in the food processing and agricultural industry. Science Bulletin BIP Biotechnology Industry Park is a specific form of industrial park specialization in biotechnology. Such industrial park usually host a cluster of the campaign that work together as a biorefinery. A biorefinery is a biomass equivalent of an oil refinery. These companies refine biomaterial and product bioproduct. Bioengineering is the science upon which all biotechnological applications are based. Bioengineering. Engineering, computer science, cell and molecular biology, microbiology, genetics, psychology, biochemistry, immunology, virology, embryology, recombinant DNA technology, genetic manipulations of the bacteria, virus, fungi, plants, and animal, often for the development of specific product. Application of the BIO technology: health, agriculture, industry, environment. Reference: Wealth is biotechnology, biotechnology in agriculture. Applications of the biotechnology in the food, pharmaceuticals, and agricultural industry. Medical biotechnology, agricultural biotechnology, environmental biotechnology, forensics biotechnology, industrial biotechnology. Type of biotechnology: green biotechnology, red biotechnology, blue biotechnology, white biotechnology. Green biotechnology defined: agricultural biotechnology is a collection of the science techniques used to import plants, animals, and microorganisms based on the understanding of DNA. Science have developed solutions to increase agricultural productivity, enhance breeders' ability to make improvement in crop and investors, enable improvement that are not possible with traditional crossing of related species alone. Plant tissue culture, fight climate change, plant genetics engineering, green bioengineering, hybrids, action biofertilizers and pesticides. Green biotechnology, use of biotechnology technique in agriculture. Vitamin A definitely is a serious problem and can cause blindness at a young age if left unstudied. Golden rice was genetically modified to produce beta-carotene, a precursor of the vitamin A that the body convert off to vitamin A. A diet includes golden rice can thus help to raise vitamin A level. Assignment: prepare a speech on future of man with the green biotechnology. White biotechnology, it is also called industrial biotechnology. It is the design of an organization to produce useful chemicals, example enzyme. Apply technology to improve food quality, role in beverage industry, machines, development of the new vaccine, development of the new process of the hiring product, role of food processing. White biotechnology, the use of biological methods to optimize industrial process applied by manufacturer of laundry detergents, include research for the new enzyme that worked under extreme conditions. This often entail modified to the enzyme of the microorganism organism for the process. Red biotechnology. The term is used in medical process. It involves the manipulations of the microorganisms to make products such as antibodies. Red biotechnology. It includes production of the medicine and pharmaceutical product for treating or diagnosis disorders, designing of the organism to manufacture antibodies and vaccine, engineering of the genetic defects through genomic manipulations, use the franchise through DNA profile. Example: production of the human insulin from non-human source, production of the for hormones like interferons, hyconins, steroids, and human growth hormones, gene therapy for preventions and control of the disease like uh, hemophilias, crystal fabrics, development of the vaccine and antibodies for rabies, use of biotechnology, development, environment, development of medicine, cloning, gene therapy, genetic testing, agricultural development. Development of the medicine with the help of biotechnology, microorganism can be used to make biological substance for human which are equally effective as natural substances. Microorganisms such as bacteria and yeast are used to make substances like insulin and interference. Treatment of disease like hepatitis and arthritis are made possible with the help of biotechnology. Drug production. Insulin. Human insulin is being produced using genetic engineering techniques known as humulins, and it is used for the treatment of diabetes, that is low sugar level in the blood. Drug production. Interferon is transmissions of the viral genome from one cells to another, and it also inhibited the cell divisions of abnormal cells. Interferon products use the recombinations DNA technology used to treat cancer patients. Interferon improve the quality of life of cancer patient. Drug productions. Human growth hormone. Science dwarfism is caused by the growth hormone deficiency, so it can be diagnosed by the HGS testing. So HGS is used for treatment of dwarfism due to the hypopituitary activities. Pouletin in 1978, genetic development humanized insulin by joining its gene with a plasmid vector inserted into the bacteria called gene therapy. The process is while a faulty gene is removed or replaced with its healthy copy to restore the normal function of the gene. For example, the disease like cancer is treated by the replacing defective gene with the hypon. Gene therapy replacing a mutated genes that cause disease with a healthy copy of the gene, inactivating or knocking out a muted gene that is functioning improperly, introducing the new gene that help fight a disease. Gene therapy. Some common ways are using fat drops in nose sprays, using cold virus that are modified to carrying LS go into the cells and affect them, the direct injection of the DNA. Genetic testing. It involves the direct examination of the DNA molecular of an organization it is used. Newborn screening. Test for the normal missing of a gene production. Carrying testing. The test to avoid transfer of the defecting gene from parents to child. Identify testing. Genetic disorder like skill cells. Genetic testing. Better drug can be obtained by the knowledge of genetic. Genetic testing can be used to detect the mutual regarding genetic disorder like cystic fibrotics, skill cells anemia, Huntington's disease, etc. Tests are also being developed to detect various cancer. Environment. Biotechnology helps to eliminate pollutants, remove waste from environment. Cloning. Cloning are organisms that are exact genetic copies. Every single bit of their DNA is identified. Cloning can happen naturally, identically twin are just one of many examples, or they can be made in the lady by placing the cell in body of organism. By placing egg cells of the petri dishes to the make embryonic stem cells. DNA of the plant is modified to enhance protections of the plant against disease, increase the quality and nutritional values of the plant, increase the yield of the plant. Biotechnology timeline 1996, first mammal cloned from adult cell. 1990s, first convictions using genetic fingerprints. 1996, develop of Affymetrix gene clips. 1997, first artificial chromosome. In December 2004, Dr. Drigits claimed in a letter to UN that cloning has successfully cloned in 13 children. Discussion: what is the society impressions of the biotechnology? What are the negativity impact that biotechnology may have? Bacterium is uncellular organism. Cellular bacteria, plural bacteria. Bacteria are prokaryotics, a member of a large group of unicellular microorganisms which have cells but lack organism and an organization nucleus. There are genetic metals in singles. Bacterium shapes. It has different shapes. Some bacteria are beneficial while some are harmful. Disease caused by bacteria, infection disease like cholera, orthorax, etc. Treatment antibodies. DNA. Doxohydronic acid is molecular that carries the genetic instruction used in the growth, development, function, and reductions of all kind known living organism and many virus. Each base pairs is attached to a sugar and a phosphates molecules. Together, base sugars and phosphates are called nuclears. Nuclear from the two stars double nucleus. DNA replacement. DNA replication is the biological process of the production two identical replacement of DNA from one original DNA molecules. This process occurs in all living organism and is the basis for biological inherent. DNA is made up of the double helix of the two complex standards. DNA replications the double helix is unwanted and each standard acts as a template for the next standard. Bases are matched to synthesize the new partner standard. DNA replication is the process of productions two identical replications from one original DNA molecules. DNA replication. There are hydrogen bonds between two standards. The first step is to break the hydrogens bond between base. This is done by the enzymes called ethylaceous. After unzipping, new bases are affected to old ones by following the base painting rules. How genes are transmitted. A gene is the unit of heredity and control information about the characteristics of an organism. Gene transfer is the transfer of the gene from one organism to another, from the donor to the recipients. Gene transfer. A part of DNA of donor is cut with the help of enzyme. A part of DNA of the recipe is cut with the help of enzyme. Gene transfer. The new DNA part is the transplanted on the recipe DNA. Enzyme DNA is the use to the resource new part of the DNA request. If you have any kind of question regarding biotechnology, you can ask me. Beside that, I want to share one of the research paper, and this research paper actually help you to understand and grow different kind of things, and which may help you to grow our understanding about biotechnology. Biological excitations of a hydrochlorofluorocarbon by a methanophytic bacteriums. Mary F. Delbart and the Robert J. Hydrochlorofluorobonds proceed as alternative to the ozone-depleting chlorofluorobonds. HFCs have been considered recalcitrants to biological degradations. We report the aerobic degradations by a mentaphronic bacteriums of 3 or 5 HCFCs and one of the three hydrofluorocarbon tested. The ability to methanase testophiums OB3B to delegate this compound dependent on the degree and position of the halogens substitution. Gas chromatography, mass spectrography, and ion chromatography were used to verify the transformations of the HCFC and the HFCS. The scoiotic release of the free chlorides and fluoride ion indicated completed mineralized by the reactions. The wide disseminations and abundance of the methanophones in the environment suggests that this organization may provide a natural sink for some of the CFS replacements compound. This is the very important aspects of the development, and if you have any questions regarding biotechnology, you can communicate with us. And if you want to share our content, you can subscribe our YouTube channel. Thank you everyone.